I'd now like to introduce Kilian Kelly from Cynata Therapeutics, ASX code CYP. Cynata Therapeutics is an Australian stem cell and regenerative medicine company that is developing a therapeutic stem cell platform technology, I hope I got this right, Cymerus, using discoveries made at the University of Wisconsin-Madison. To tell us more about this important work, Kilian, over to you.
Thank you very much, David, and thanks to everybody for joining today. So yeah, I'm going to tell you a little bit more about Cynata Therapeutics. We can move to the next slide, please. So Cynata is a clinical stage biotechnology company based in Melbourne. As David said, we are focused on the development of our proprietary platform technology, which we call Cymerus. And what that is used for is to enable the scalable manufacture of mesenchymal stem cell or MSC-based therapeutic products. And those products in turn can be used to treat a wide range of serious medical disorders with unmet needs. So this is a unique manufacturing platform. And the reason I say that is our platform allows us to make an essentially limitless number of product doses from a single donation from a single donor. And that really sets us apart from other conventional cell therapy manufacturing processes. And I'll tell you a bit more about that in a moment. So over the past few years, we've generated strong safety and efficacy data using our products in a range of preclinical and clinical trials. We now have several active clinical programs, including acute graft versus host disease, diabetic foot ulcers, osteoarthritis, and renal transplant. And collectively, those conditions represent a pretty huge addressable market. So it's estimated at about $28 billion annually in total. So there's clearly enormous opportunities for us here, even if we're only successful with a subset of the products that we have in development. The company's well funded at the moment. We have enough cash in the bank to complete the ongoing clinical trials. And it's also worth mentioning that 2 out of those 4 clinical trials are actually fully funded by external partners. So we can move to the next slide, please. So as I said, our currently active clinical program includes a total of 4 clinical conditions. And I'll just briefly mention that we do have other indications that we have preclinical data on, but we're not currently in the clinic. But in the clinic right now, we have 2 programs that are sponsored and funded by Cynata. And they're acute graft versus host disease, which is in a phase 2 clinical trial, and diabetic foot ulcers, which is in phase 1. Then our partnered programs include osteoarthritis, which is partnered with the University of Sydney and funded under an Australian government NH&MRC project grant, and that's in phase 3. And then we have a recently approved new trial in patients who have received a kidney transplant, and that's partnered with Leiden University Medical Center in the Netherlands, and they are funding and managing that trial. Next slide, please. So some of you may be wondering, what are MSCs and why are we interested in them? So these are cells that actually exist naturally in all of our bodies in fairly small numbers. And they're known as the sensor and switcher of the immune system. So they promote an immunomodulatory and immunoregulatory environment. And really what that means is, in plain English, is they calm down the immune system and they basically keep it in balance. And the reason that that's important is that the immune system, we have a lot of inappropriate and excessive immune system responses to various triggers. And those responses underlie quite a wide range of medical diseases. So a product that is able to balance and manage that immune response has many potential therapeutic applications. And that's really where MSCs sit. Next slide, please. So just to explain what is unique about our manufacturing platform, a conventional stem cell manufacturing process involves taking a tissue donation, for example, bone marrow or fat or some other source of tissue, and then isolating the MSCs from that tissue donation, and then growing those cells in the lab, which is a process known as culture expansion. And the reason that's necessary is you don't get many cells from each donation. So for example, you might get about 20,000 MSCs from a bone marrow donation, but you may require upwards of 1 billion cells to treat a single patient. And of course, in most cases, you're interested in treating thousands or even millions of patients. So you can see the amount of expansion that's required to turn 20,000 cells into this number of cells that you need. And the reason that this is a real problem is that it's well established that MSCs actually change when they undergo this culture expansion process. So you can generate quite large numbers of cells, but what you'll find is they start to lose the properties that you're trying to harness and they become less effective. And that means you need to get new donors on a regular basis. But that's a problem as well, because it's very well established that there's a huge amount of variability from one donor to another. So essentially, we have two sources of variability and potential suboptimal functionality in MSCs produced in this way. And that's exactly what the Cymerus process was designed to overcome. So we use a relatively recently introduced technology known as induced pluripotent stem cells, or iPSCs. And I'm sorry to use so many different acronyms for different types of cells, but I hope you can bear with me. So iPSCs have the capacity to reproduce themselves essentially indefinitely. So that means we can produce an effectively limitless number of cells from a single donation from a single donor. So we completely avoid that problem of inter-donor variability or variability from one donor to another. We then have a patented process to turn iPSCs into MSCs. But because we get our scale at the iPSC stage of the process, we don't have to extensively expand the MSCs once we form MSCs. So we also avoid that problem where you get changes in MSCs as they undergo extensive culture expansion. So in summary, the Cymerus process allows us to overcome the two fundamental challenges with large-scale manufacture of cells of this type. So next slide, please. So I won't dwell too long on this slide, but this study was conducted by Monash University, and it found quite clearly that there are differences in MSCs from different sources, including Cymerus MSCs. But importantly, the study found that Cymerus MSCs exhibit less batch-to-batch variability and less intrapopulation variability. And what that means is variability within a batch. So they have less variability between batches and within batches compared to tissue-derived MSCs. And that's what we would have expected. And this really underlines the premise that we are pursuing here. Next slide, please. So we have a strategic partnership with Fujifilm. It's a strategic manufacturing partnership. So they are our manufacturing partner. You may or may not know that Fujifilm is a world leader in the manufacture of cell-based therapies. Of course, it's a well-known company for other products as well, but they are a leader in the manufacture of cell-based therapies. So this really puts us in a very strong position to kind of grow our own manufacturing capability as we move further into commercial scale. Next slide, please. In terms of our clinical trials, our first completed clinical trial was in a condition called graft versus host disease. And it's worth noting that this was actually the first completed clinical trial of an iPSC-derived product of any kind anywhere in the world. So it was quite notable. Graft versus host disease is a truly awful condition. These patients have terrible symptoms, and unfortunately, the majority of them end up dying within 1 to 2 years. The patients in our trial were treated with steroids but had not responded, and that was the only available treatment at that time. And patients who fail to respond to steroids have a really poor outlook, and they're not expected to improve spontaneously. But we treated 15 patients. 13 of them showed a response of at least 1 grade, an improvement of at least 1 grade, and 8 out of 15 showed a complete response, meaning the condition resolved entirely. The 2-year survival rate was 60%, which is very high in the context of these patients. With standard of care treatment, when they fail to respond to steroids, the reported survival rate is around about 20% after 2 years. So these results were really encouraging, and they form the basis for us to move into a phase 2 trial, which on the next slide is summarized. So this trial has just recently opened for recruitment. This is a randomized trial in 60 patients. It's a global trial. It's already got approval in Australia and the US. We expect to complete recruitment in late 2024 with results available around about the second half of 2025. And of course, we're really excited to build on those fantastic results in phase 1. Next slide. So our diabetic foot ulcer trial, this involves a novel wound dressing where the cells are actually seeded on a dressing that is put directly onto the wound. Now, diabetic foot ulcers are chronic wounds that occur in patients with diabetes. They are very difficult to treat and they often don't heal, and they're then subject to all sorts of complications, which ultimately can lead to amputation of the foot or the lower limb. So it's a very serious problem. In this trial, we're randomizing a total of 30 patients to either standard dressings or the novel MSC dressing. We expect to complete recruitment by the end of this calendar year with results in the middle of next year. But on the next slide, we do have some initial data from the first 6 patients, which is really very encouraging. And what you can see here, this is the percentage of the ulcer that has healed after 28 days. And you can see that in the light blue line, in the active MSC group, there's a substantially higher rate of healing after 28 days compared to standard of care. So if we see those kind of results once the whole trial is completed, that would certainly put us in a strong position to move forward. Next slide. So our osteoarthritis trial, as I mentioned, this is partnered with the University of Sydney. So they're conducting this trial, but using Cynata's cells. And it's important to say that Cynata has the commercial rights to use the data from this trial, and that's very important. The trial is being funded by the Australian government under an NH and MRC grant. We've already recruited more than 300 patients in this trial, and we're expecting to complete recruitment around about the end of this year. The patients will then be followed up for 2 years. And the reason for following them for so long is we're looking to show a disease modifying effect here, which means that we're actually not just trying to improve the symptoms in the short term, but actually slow down or stop the degeneration in the knee joint. And if we can show that, if that's successful, that would be an absolute game changer for the treatment of osteoarthritis, because right now there are no treatments that do that. All of the available treatments just manage symptoms, and ultimately patients end up needing to have surgery, including potentially a knee replacement. So this is a hugely exciting trial in an absolutely huge problem worldwide and a huge market opportunity. Next slide. And so finally, our renal transplant trial, this has just been approved by the regulatory authority in the Netherlands. And as I mentioned, this is partnered with the Leiden University Medical Centre. It's a similar sort of model to the osteoarthritis partnership with Sydney, in that LUMC will be managing and funding the trial using Cynata's cells, but Cynata will have the commercial rights to the data. So the idea of this trial is patients who receive a kidney transplant normally have to take anti-rejection drugs for the rest of their lives, but those drugs are really toxic. Some of them can dramatically increase the incidence of serious infections and cancer, and ironically, they also cause kidney damage. So these drugs are very problematic. And what we're aiming to do in this trial is that after the transplant, patients will receive MSC treatment, and then the dose of those anti-rejection drugs will be significantly reduced. And if we can show that that does not cause any problems, such as an increased rate of rejection, then that would allow us to potentially reduce or eliminate those drugs moving forward. And that would really be expected to change the outcomes in these patients in a very positive manner. So as I said, this trial is just about to commence, and it's another very exciting one that we look forward to getting results from in due course. And in the interest of time, I think that's about all I've got for you. On the next slide, we do have the standard disclaimer, and then the following slide is just the closing slide. So I'd be very happy to take any questions, David, that may have come in.
Thanks, Kilian. Great presentation. We do have a number of questions that have come through so we'll jump straight to them. How does Cynata differentiate itself from competitors in the field of regenerative medicine?
Right. So it really is based on how we manufacture our cells. And as I mentioned in the presentation, a typical way of producing cellular therapies is to take tissue donations and basically isolate the cells from those donations. It might be bone marrow or blood or fat or something like that. And that means they have to get this ongoing supply of new donors. And that is associated with a lot of variability, but also logistical challenges, as you can imagine. If you were making these products if we were on the market for osteoarthritis, for example, potentially treating millions of people every year, imagine if you need, new donors every couple of days to try and supply that. It would be extremely logistically challenging. But I think more importantly is the variability that introduces. And we completely avoid that by being able to make all of the products that we need from the same original donations. We have a cell bank that has an essentially limitless capacity. So we'll just keep using the same starting material time and time again. And that really is a game changer.
What about the size of the prize? How big's the market that you're playing in, and how's it evolving? Is it a growing market? Is it a stagnant market? Where do you see the opportunity?
Yeah, so look, the market opportunities are enormous. So even if we look at the 4 conditions that we're already in clinical trials for, which I should emphasize, that is really a small subset of the potential, really, but that's what we're focused on right now. But those 4 markets on their own add up to an estimated $28 billion US annually. And even if we look at the very smallest of those markets by a long way, which is graft versus host disease, on our slide there, it looks tiny, but it's actually $600 million US annually is the addressable market. So the potential commercial opportunities here, even if we're successful in just one of these conditions, the commercial opportunities are absolutely enormous. And in terms of whether they're growing, I suppose it's good and bad news. They are growing, which is bad news, I guess, for humanity, in that most of these degenerative medical conditions are becoming bigger and bigger problems as time goes on. Diabetes is becoming a bigger problem. Osteoarthritis is becoming a bigger problem. So that's a huge challenge for society, but it's a huge opportunity for companies like us that have potential solutions to these problems. And I don't think there's any doubt that these markets will continue to grow.
Now, there was some mention in some of the previous announcements about a strategic review.
Mm-hmm.
What's the long-term vision and the strategic direction now you've been through that process?
Yeah, so that's correct. We conducted a strategic review in July, and the outcome of that really is that we are focused on those 4 clinical programs for now. And our direction, really what we're aiming to do, we have an out-licensing business model. So as you may be aware, the resources required to take a pharmaceutical type product all the way to market and then to actually market that, it requires enormous resources. And that's really what big pharma does very well. So what we're aiming to do is to generate the data that shows that these products are safe and effective, and then out-license them to larger partners. And what that means, though, really is that our potential revenue is not necessarily too far into the future. I mean, people often think, oh, well, you're in a phase 2 trial. You could be years away from getting this product on the market. But the reality is that we could be generating serious revenue way before that by out-licensing the product. And that really is what our aim is.
So maybe that brings me to my last question that's just come through. Can you highlight key priorities and objectives for the coming year? What should investors be focusing on?
Right. So we're seeking to complete recruitment in 2 of our clinical trials around about the end of this calendar year. And that's the diabetic foot ulcer trial and the osteoarthritis trial. And of course, completing recruitment is a major milestone, because that then pretty much locks in the timeline to when we're going to get results, which is what everybody is focused on. So there are 2 major milestones coming up. In addition to that, we should be commencing the renal transplant trial before the end of this calendar year. And then looking a bit further forward, in the middle of next year, we should be getting results from the diabetic foot ulcer trial. Towards the end of next year, completing recruitment in the GVHD trial, and then getting results from that trial around about the second half of 2025. So we've actually got a lot of milestones coming up.
As you say, a lot of milestones coming up. So clearly a company to put on the watch list and just follow with interest. So Kilian, thanks for your time. We look forward to following with interest.
Thank you very much, David.